Hello everyone, a very warm welcome to you all and thank you for joining us for today's presentation. I'm John Norman, Head of Marketing here at Optibrium and moderator for today's webinar. And it's great to see so many people joining us for this presentation on eSim3D, electrostatic field and surface shape similarity for ligand-based drug design. And for today's webinar, I'm delighted to be joined by Dr. Ajay Jain, founder of Biopharmics, and Matt Segall, who is CEO here at Optibrium. Matt, I will now hand things over to you.
Thank you very much, John, for your kind introduction. It's a great pleasure to see so many of you here today again. And it's also a great pleasure to welcome Professor Ajay Jain to our webinar. Ajay is professor at UCSF and the founder of Biopharmics. Ajay's worked in applied computer science in many domains, beginning in the defense industry, looking at both target detection and recognition, continuing in speech and language processing, and ultimately leading to the bulk of his postgraduate career in computational chemistry and biology. With specialties in machine learning, drug discovery, and bioinformatics, Ajay's current work is geared towards applications of computation to drug discovery, with an emphasis on making use of multiple methods and data types, exploiting synergy between protein structural data, chemical and target annotation information, and ligand binding affinity data. So many thanks for joining us today, Ajay. I just thought before we get started with the main presentation, I was intrigued by your very multidisciplinary background. And I guess nowadays, it's very, very common to see people moving from computer science and machine learning into drug discovery, but that's a fairly recent trend. And I think you were one of the pioneers in making that shift. And I just wondered if you could share with us a little bit about what attracted you to the field in the first place.
Sure, Matt, and thanks for hosting this webinar. This should be interesting. Yeah, my intentions were fairly random. Back when I started an undergraduate, I really didn't know what I wanted to do. And so I did a degree in biochemistry and I did a simultaneous degree in computer science, not really understanding which field I wanted to pursue. What became clear, though, was that the pace of research in biochemistry was frustrating for me. You have an idea and it's 6 months, 2 years, something like that, to try to figure out if you're half right or half wrong. With computer science, generally speaking, you can work as fast as you think. Given enough pizza and coffee, if you're willing to work, your pace is only limited by your ability. I also started working in the defense industry as sort of a summer job back then. This was during Cold War days, and that really presented me with a lot of interesting computational problems. So then I ended up pursuing my PhD in computer science at Carnegie Mellon. After I wrapped that up, we were still actually in the middle of the Cold War, actually towards the end. And I stayed in the defense industry for a little bit of time. All the while, though, sort of missing physics and chemistry and biology, because there's a lot of depth there that you don't get in a highly applied area like target recognition in sort of missile guidance systems and so forth. Well, then, sort of, I guess, conveniently or not, peace broke out at the sort of end of the 80s, and I actually got a job ad from a small biotech startup in South San Francisco. They were looking for somebody who was formally trained in biology and chemistry, but who had a PhD in computer science, who specialized in machine learning. As you said, I mean, at that time, as far as I knew, I was the only person in North America with that weird set of qualifications. And so I went out and interviewed and joined Ares Pharmaceutical shortly thereafter, and have really enjoyed the blending of interesting algorithmic ideas with understanding physics and chemistry and how biological systems work.
Wow, Ares Pharmaceuticals was definitely forward thinking for their time, and I guess I can share your experience of coffee and pizza fueled coding sessions from my past as well. So thank you very much. That's absolutely great. So without further ado, I'm going to hand over presenter duties to you. and very much looking forward to your presentation.
All right, thank you, Matt. I assume everybody can see my screen. I want to talk about work that's been done now, actually quite recently, with me and my longtime collaborator Ann. This is based, though, on much earlier work in the molecular similarity field dating back to the late 1990s. What we've always thought is that thinking about molecules from the perspective of how protein views them, i.e. looking at the molecular surface, is the right way to calculate things about similarity, or even calculate things about how they might dock, or what have you. I want to talk about three different areas of application for molecular similarity in 3 dimensions, with the key idea being that you can actually support drug design fairly aggressively with purely ligand-based methods, very much like you do with structure-based methods. So virtual screening is sort of the obvious thing. If you have a couple of ligands that you know have some activity against your target, you can screen large databases for new ones. Key issue here is how well you're able to identify actives, ideally with very low false positive rates, because the libraries that we have now for virtual screening are very large. Something that I think everybody has cared about for a long time, but has had less confidence in, is the ability of ligand-based methods to actually predict poses accurately. And we've actually done some very large-scale testing of pose prediction, which we'll get into towards the end. Underlying multiple ligand alignment, and the reason I think that it's a challenging problem, is that this pose prediction, purely based on ligand structural information, is quite challenging. We've made quite a bit of progress there, and we'll talk about that as well. Okay, so there's sort of a number of different ideas in the field of molecular similarity, and I think many of you will be familiar with them. Obviously, the Rock's idea of using atom-centered spherical Gaussians in order to compare molecular structures, molecular shape, is very widely used and appreciated. The sort of alternative view that we think is a bit more physically realistic was initially implemented in SurfLex-Sim, the morphological similarity approach. Now we've actually generalized that into the eSim3D approach, which I'll talk about today, including the differences from the prior approach. Obviously, there are pharmacophoric approaches and electrostatic field comparison methods, other things. But for the moment, let's just focus on the atom-centered Gaussian contrast with surface comparison. So if we look in the center here, we've got 2 different inhibitors of trypsin. One's benzamidine, one is aminomethylcyclohexane. This is how they actually look relative to one another when they're bound to trypsin. You can see that atom centers don't actually align with one another in this experimental crystallographic models aligned on the trypsin backbone. If you look at the optimal superimposition of these 2 molecules using atom-centered Gaussians, using the Rock's approach, what you're doing here is you're trying to maximize the overlap integral of the sets of spherical Gaussians for the 2 molecules. And over here, we're seeing a slice horizontally in the plane of the slide through that overlap integral. And what you see is sort of the atomic structure of the 2 molecules, with a great deal of the important piece literally residing on top of atom centers. This alignment qualitatively doesn't look really like how things appear when the biology is happening, when you have these things bound to trypsin. What we do with SurfLex-Sim and also with eSim3D is look at the molecular surface and compare differences between ligands based on that. So when we look at the optimal alignment that one gets using the surface-based approach, and then look at the field that's implied, what we're seeing is that we're trying to maximize the overlap, the congruence of molecular surfaces. And we see qualitatively, again, an alignment that makes a lot more appealing relationship with what we see experimentally. So how do we do that? I'm not going to spend too much time on this. The paper is very detailed in how it's done. The basic idea is, given some target ligand, we're going to place molecular observer points around the ligand. We'll do this in 3D, and I'll show you a picture of that. At each one of these observer points, we're going to calculate the minimum distance to the molecular surface. That's going to capture molecular shape. We're going to calculate the coulombic energy of having brought a charge from infinitely far away into that position. And so we're going to get an actual electrostatic field measurement that's dense around the entirety of the molecule. We're also going to look at a representation of hydrogen bond donors and acceptors that's directionally specific. So if we have this observer down here, the distance to the nearest hydrogen bond donor is a little bit over 4 angstroms. And this angle here, sort of ideally you want the observer to be looking straight down the barrel of the NH over here. This angle is a little bit less than that. The nearest acceptor is quite far away, and the angle is not acceptable really to make a hydrogen bonding interaction. So we're capturing these 6 pieces of information at every one of these observer points, and the game now is going to be when we bring another molecule in to align to this in a conformationally flexible way, we're going to want to minimize the difference in these measurements. We're going to do that through Gaussian functions. So here in 3D, we're showing you the same molecule, the full set of observer points in yellow spheres. So 2-pyrrolidone is our target molecule. Some would refer to this as a query. And here I'm showing you the optimal alignments of a few different small molecules. It shouldn't be terribly controversial that cyclopentanone and pyrrole both make sensible alignments, and that they have relatively high similarity on a scale from 0 to 10 when measured against 2-pyrrolidone. Obviously, as we get into a purely hydrophobic subject molecule, benzene, we get quite low similarity because electrostatically benzene and 2-pyrrolidone are very different. Clearly, if we vary the charge in different ways with an amidine or carboxylate, we get slightly higher scores than benzene, which may or may not be reasonable according to your intuition, but they're still less than, I think, the appropriately higher scored molecules. So in order to do all of this and do it quickly, one of the major changes from our older methodology for doing molecular similarity is that we do conformational search before we run the eSim3D alignment protocol. So each of these things, while they're fairly rigid, there are some conformations that vary. But with real molecules, here we have tetracycline over here. Obviously, we get very significant conformational variability, even in what looks like a fairly rigid ring system. We've developed a technique called ForceGen, which we published a few papers on now, that's applicable both to small molecules and to macrocycles. It's very fast and extremely competitive. We would say actually definitively superior in terms of both speed and accuracy on very large benchmark data sets. Those are discussed in the second paper down here, published with our colleagues from Merck in 2019. I'm not going to go through the details of ForceGen or the benchmarks. I want to encourage you to look at the papers. What I do want to point out is that under the hood, what's happening is we're beginning from a 3D representation of each molecule and producing a compressed database of conformers for the many molecules that are in this database, in this set over here. The reason we need to do the compressed database is that even with solid-state hard drives now, the eSim approach is so fast that if we were reading from individual multi-mol2 files, it actually became a huge bottleneck. All right. So how do we do validation? How do we know that this is actually working at all well? Well, the sort of de facto benchmark that people have come to use is DUD-E from John Irwin and Brian Choquette, which was designed as a docking benchmark across a very large number of targets. where each of the targets has a set of active ligands, a fairly significant number on average, and has property matched decoys from the zinc database that are known not to look a lot like the actives. There's some peculiarities in using such a benchmark that's designed for docking for ligand-based methods. Because one needed to avoid having false positives in the decoys, 2D similarity was used to prune obviously similar molecules from the decoy set. So if you look at any 2D similarity method that's not insane, it'll work pretty well, apparently, on this data set. So really for 3D methods, you can use the DUD-E set to assess 3D ligand similarity virtual screening performance, but you can't really do it for 2D methods and expect to believe the results. So giving you a couple of examples here, there were a couple of nice papers published in the past 5 years benchmarking many different ligand-based methods using DUD-E, using the full set. So eSim, which we published a couple of years ago, these are our results. The other results in here come from the respective papers that were run by other groups. What we're showing are receiver operator characteristic area under those curves. Okay, so 1.0 is what you want. 0.5 is exactly random. Below 0.5 is actually worse than random. And so these are fairly typical results that we get. The DUD-E target for carbonic anhydrase is a trifluorosulfonamide here, trifluoromethylsulfonamide, which is a very odd thing to use for ligand similarity. But it happened to be the ligand that was bound to the protein structure that John Irwin and colleagues picked for DUD-E docking. Here, this is an example of an active molecule, a more typical looking carbonic anhydrase inhibitor. This is the predicted alignment in a fast screening mode of this particular active molecule against the bound pose of this carbonic anhydrase prototypical target. You see it's reasonable. We get the warhead in the right place. Both of these things interact with a catalytic zinc. eSim's ROC area is 0.92. Everybody else does random or worse, essentially. We get a score that is maximized at 10, typically doesn't go below 2 or so. We get a quantitative estimate of strain, having done rigorous conformational search before the eSim calculation. And this score is bigger than 99.9% of all the decoy scores, which is why we get such a good ROC value for the area under the curve. More interesting target. Here we've got PARP1, interesting cancer target. Here's the query molecule, if you will. The warhead is on this side over here. This is an example active, and again, we're seeing very high performance for eSim, decent performance for some of the other methods. Again, this is a low strain conformation of the active in particular. We get a bit of a lower score here, but we're still bigger than 90%, 96% of the decoys. A couple more examples here. We have a kinase, a thymidine kinase over here, where we've got a very typical looking inhibitor, early inhibitor of this kinase, typical active. And again, we see the same kind of performance. I wanted to show you one case here, FAK1, where ROX did significantly better than eSim. This happened in, I think just this one case. It might have been a couple of other cases, but across the board, the eSim approach really does very well. So if we look at, I'm showing you a big table here. This is across all 102 DUD-E targets. At the second fastest level of screening, the eSim approach does better than ROX color by a p-value of 10 to the minus 9. Okay, so all of the eSim variations from the very fastest to the most thorough screening mode do better than ROX color. ROX color and ROX shape do substantially better than all of the other approaches that people had published. where people were really trying to emphasize speed in some of these cases. But the average performance of these methods is so close to random that it's hard for me to understand why people sort of decided to publish. These are sort of negative results in my view. Using a 20-year-old method, just computing molecular imprints, using sort of like SIM, again, an old technique, you can achieve better results than all of these methods at speeds of over 200,000 molecules per second. ROCs, non-GPU ROCs, now single core, single CPU, typically runs at about 50 molecules per second, having pre-search things with Omega. eSim in its fastest mode is about 5 times faster. In its slightly more accurate mode, it's roughly the same speed, single core, single CPU. Okay. One of the things that we were interested in, though, scientifically, is to what extent can you take advantage of multiple ligands and see what the variation is using different ligands as your query, or perhaps using a multiple ligand alignment or the bound crystallographic poses. So we've constructed a new benchmark we call DUD-E+, which contains 92 of the original targets, each of which where we've automatically selected 5 exemplar ligands that have known bound configurations. This was published in 2019. The data set is publicly available, and we'd encourage people to take a look at it. For LCK, another kinase, these are the 21 example proteins that have been aligned using a local binding pocket similarity approach that's closely related to the eSim method. Here we see the collection of ligands. So what we did is went through and, again, automatically using a k-means clustering approach, select 5 ligands for each of these targets. For LCK, what we see are these 5 ligands over here, which coincidentally included the original DUD-E ligand, which, as you can see, is a highly atypical kinase inhibitor, along with several more, much more typical looking inhibitors. This is what their set of bound poses looks like when they're superimposed. And you can see that this now reaches both towards solvent on the left-hand side and internally into the kinase for when you see a DFG conformational variation. If we use now this multiple ligand target as the query for eSim, we see a very significant improvement over when we use just a single atypical kinase inhibitor, where we do just a bit better than random. We start doing much better, 0.84 using the joint superimposition. It turns out that across the entire set of these 92 targets, we see the same behavior, that going from the original DUD-E ligand to sort of cheating and saying what would be the best single alternate if we knew the right answer ahead of time, we do better, obviously, than with the original target. Using the joint set of 5 alternates without actually knowing which one's the best, or if even there's one that's pretty bad, consistently does as well or better than knowing what the best single alternate is. So when eSim aligns to a multiple ligand query like this, what it does is it sort of treats the different choices as a pick and choose. So if when I align my subject molecule in here, it likes this donor down here, it's going to say, well, I want to look like that. And if when I align the left-hand side, I like this acceptor functionality over here, I'm going to choose to like that. So you get sort of some synergy from the different ligands. Just to give you an idea of some of what the timing looks like for this carbonic anhydrase example, using just an 8-core machine and looking at 500 actives and 3,000 decoys. This is processed at about 950 molecules per second. We get an ROC area of 0.87 using the PFAST level of screening. If we use the fastest level of screening and we run this using the 5 ligand superimposition, We only go about twice as slow, even though we've got 5 ligands to calculate our similarity against. So you don't pay a linear cost in having n ligands in your multiple ligand target. For the PARP target, we see a similar effect, larger set of query molecules means it goes a little bit slower, so 800 molecules a second, dropping down by a little bit more than a factor of 2 using the 5 ligand superimposition. Just to give you an idea, on a 4-core laptop, actually the laptop that I'm showing this presentation on right now, just a Dell XPS 13. Again, it's very fast, 240 molecules per second for a single ligand query, and about 3 times slower for a 5 ligand multiple ligand query. Okay. So why is it that consistently across this very large data set, again, that we didn't design the data set, we didn't choose the targets, we took it as it came. We think that's a fair way to use a benchmark. Why do we think that eSim does better? Well, we're going to assert for the second that it's predicting the right thing for the right reason. Recalling back the fundamental difference in the representation of what molecules look like and what should be similar when you compare them to one another. So we've gone through the virtual screening. We've shown you the statistics across a large number of targets. Now let's go through this question of pose prediction, because I think that's the way to ask whether you're doing something for the right reason. So if you're getting high similarity scores and you're doing so by predicting optimal poses that actually match with crystallographic experiment, that should give you some confidence that you're predicting things for the right reason. So we took, so one little detail here, which I think is important. There are ligands that can bind competitively and actually be highly similar to one another without having really any direct atom for atom correspondence. So if we look at these 2 different ligands, which both bind the estrogen receptor, these are their bound configurations. They occupy exactly the same volume, but they share almost no direct atomic overlap. It's really only over here. Other than that, the scaffolds do not align. And you might actually notice some more than superficial similarity at the scaffold level between these 2 molecules, but they actually bind in opposite directions in the pocket. So again, anything that's sort of atom-based will get this pose prediction wrong, because it's going to try to superimpose the benzene on top of this benzene and this functionality on top of this functionality. That turns out not to be correct. Okay, so we took the DUD-E plus set and we generated near 400,000 pairs of molecular alignments, and we restricted ourselves to what we consider to be tractable alignments. There are cases where you get enough overlap between 2 poses, 2 different molecules, that, you know, if a similarity method is working pretty well, it should be able to get that right. There are other cases where you'll have 2 ligands which only overlap by, say, about 10% or up to a third if you're looking at volumetric overlap. We don't think these things are tractable by similarity methods, and so we're only focusing on the things that are tractable, and there's details about that in the paper. So here we've got a non-macrocyclic target for estrogen receptor 1. This is our subject molecule. This is an environmental estrogen agonist, and what I'm showing you over here is the top scoring pose family from eSim of this guy here aligned onto this guy in its band configuration. And you can see that the best pose out of here is less than 1 angstrom RMSD away from what we know the bound configuration of this ligand actually is in estrogen receptor. On the left here, this is the actual alignment that we get from molecular similarity. We have a way of visualizing. We see these green sticks. The orange sticks are indicating that the electrostatic field is also congruent between the molecules, and the red and blue sticks are large if the donor and acceptor functionality match up. Over here, where we have this protrusion on the target molecule, we don't see any sticks over here because there's nothing that's matching. All right, so what do we see? On this roughly, again, 400,000 pairs over the entire set, within the top 20 predicted poses, what we see is 80% of the time we see something within 2 angstroms RMSD. So that's pretty good. For those of you who remember the docking literature, even for a doctor, you'd be pretty happy with pose prediction at that level of accuracy. Now, this splits pretty sharply between the easy cases, where we have a lot of direct atomic overlap, where the scaffolds are similar, where we see over sort of 93% success, and then the hard cases, where we see different scaffolds, where we're down close to 70%. Now, if we want to ask, what about the very top scoring pose? Now, this is a much harder problem. There we're seeing more like 65% overall. Really pretty good, over 80% when you're looking at similar scaffolds, but down slightly below 50% when you're looking at significant scaffold variation. So if you have 2 molecules that have quite different scaffolds, you should really expect to look at a set of top scoring poses to get to see the one that's correct, that's giving you the correct alignment. With Steve Johnson at BMS, Steve is a very familiar expert ROCks user, and he ran ROCks in a number of different ways, trying to find the optimal performance for ROCks on predicting poses. And here I'm showing you the cumulative histogram for eSim in the best pose of top 20. Again, roughly 80% accurate. ROCks is down just at about 60%. So very, very significant performance gap that even increases at the 2.5 angstrom level. Top rank pose performance differences are still very substantial. This was true across all the different targets. The p-values here were very extreme, like 10 to the minus 10 or 20. Here we're showing you a heat map of all 400,000 alignments, where we have the RMSD for eSim on the right, the x-axis, RMSD for ROCks color on the y-axis. And you can see that ROCks produces some really crazy alignments in cases where eSim is getting sub 1 angstrom RMS deviations. Okay, so what's the implication of that? If you can start to get decent predictions of poses, I hope you believe at least that quantitatively pose prediction seems to be pretty respectable. And I think that's a good reason why the method seems to work pretty well in the virtual screening arena. But because we're able to predict poses reasonably well among sort of a top set of top scoring poses, there's reason for optimism in looking at multiple ligand alignment. So we have a command called multisim inside the E3D module, where first we're going to have to do a ForceGen to do conformational search on these 4 benzodiazepine ligands. We get an SFDE out of that. The multisim command will then generate a mutual alignment of these molecules here. These are pretty rigid, so the conformational search just takes about a second, and the multiple alignment takes just about a dozen seconds more. So very quickly, you're able to get multiple alignments and really browse through. This is not the top scoring solution at this search depth. It's the 3rd ranked solution. We've actually published a few papers that make use of benzodiazepine ligands, and this alignment here, even though it doesn't sort of make a direct scaffold alignment of this molecule down here to the other 3, is actually very predictive of the behavior quantitatively binding affinity of other benzodiazepines, including things like carbolines and pyrazolopyridines and so forth. Okay, so it's pretty fast. If you're interested in looking at the alignment of a large number of molecules, if you have a big series, you can generate an initial core alignment, as we've done in this slide here, and then use that as the subject of an alignment operation for the remainder of your series. Here we're looking at an alignment of 147 ligands. I'm showing you just 136 because some of them have very large substituents that obscure the sort of coherence of this multiple ligand alignment. It takes about 46 seconds, again, on the 8-core machine to do the conformational search here, just 3 seconds to generate this alignment. And if you look very carefully and click through these one at a time, all of these are very reasonable, but you would find them acceptable if you knew about the SAR here. Okay, so wrapping up with the sort of formal presentation here, what we have is a novel method just introduced a couple of years ago, and you can use it for virtual screening, post-prediction, and also for informal SAR analysis, and also to drive affinity prediction for other methods. We have a method called Quanza, but even for generating alignments for physics-based methods, if docking is unable to, the eSim approach is very capable of producing the kinds of alignments you need to kick off physics type simulation prediction for affinity. The key features of the approach are that it's surface-based and it's insensitive to direct atomic correspondence. We consider the congruence of Coulombic fields. There's a lot of detail in the paper about using, for example, a Coulombic field alone or using molecular shape alone and so forth. It turns out that just using electrostatic fields does pretty well in terms of virtual screening performance, not nearly so well in future experiments that we did in predicting poses. That's where you really do need to get into directional hydrogen bonding and have an accurate representation of molecular shape. We don't need a lot of knob twisting to get sensible results. Everything is built upon the ForceGen conformational search method. I want to emphasize that in the more thorough search modes, eSim itself actually does local conformational optimization, both using internal coordinates and all atom Cartesian optimization for the most accurate modes. So the ForceGen conformer pool that you start with isn't restricting you from moving things around if you need to. We've got benchmark performance that I think is actually pretty convincing in terms of the DUD-E set, with all the limitations that, there's no perfect benchmark out there. I think this is probably the best one that exists for measuring these things for lots of methods. The DUD-E plus set, I think, is better because it gives you an idea of how different query ligands affect your results. Post-prediction results, we don't know how any other method but the ROCks approach compares. We'd love to see other people use this. All of these benchmarks are available on the lab website. Multiple ligand alignment, it's on the list of papers to write. We've actually done some formal benchmarking here, and we want to get that paper out hopefully before late fall this year. I want to thank my longtime collaborator, Ann Cleves. Many of you probably know she and I are also married. Steve Johnson has been a huge person for us in collaborating on helping us think through methodology, but also on doing some benchmarking and being a critical eye. We've had NIH funding over many years. The full Surflex platform includes many modules. We've constructed a single module out of blending together the Surflex tools, ForceGen pieces with the eSim pieces into a module with eSim3D that we're working with Optibrium on, which is the reason that I'm talking with you today. And Matt, I guess over to you for questions.
Thank you very much, AJ. Thanks for a fantastic talk. And it's astounding that the lengths that you've gone to benchmark and validate the methods that you've just described. And I would encourage all our listeners to look up the papers and see the sophistication of those benchmarks. I'm just going to grab the screen back again. AJ has just given you a little bit of a teaser for what's to come. And so we've been delighted to be working with Ajay and Ann for a few years now with the eSim3D methods that he's just described. And we're delighted to let you know that we're going to soon be announcing a new module for StarDrop that will bring these methods to StarDrop so that you can do confirmation generation using ForceGen, virtual screening using eSim, and of course the multiple ligand alignments as well to generate binding hypotheses against which you can actually perform your virtual screens as well. So not just relying on a single active conformation. So it's a really exciting time for us. We'll be doing some demos of that real soon now, and doing some more webinars when that's released so you can see some examples of that in action. And as AJ said, we're ready to move on to the Q&A session. I can see that there are several questions already coming in on our panel here, so that's fantastic, and thank you all. If you haven't asked a question yet, please feel free to do so. Just type it into the question panel there. It'll pop up on our control panel, and we'll get through as many of those as we can in the remaining time. And so without further ado, I'm going to pose some questions to you, AJ, and here's a great one. It says, first of all, thank you for an enjoyable talk. But then also, how much SAR do you have to know ahead of time? For example, do you have to know that the critical binding interactions, for example, directed hydrogen bonds to pick out the real actives versus artifacts?
The interesting thing about the eSim approach is you don't have to have any activity data at all. What you need to have are at least one active compound. And if you're going to do, for example, a multiple ligand alignment, you would prefer to know that those ligands are actually mutually competitive. So for enzyme inhibitors, that's easy to figure out. A lot of times with some of our collaborators where they're doing complicated functional phenotypic assays, you might not know if your ligands are actually competitive. And in those cases, actually, you can use multiple ligand alignment and scores that come out of eSim in order to assess the probability that the ligands are in fact competitive. If you get high eSim scores, you can compare those to the distributions of known cases where you have different scaffolds that bind the same site, and get some confidence that the ligands are in fact competitive with one another. For our Quanta approach, which eSim sort of feeds into, you use actual quantitative binding affinity data, and typically we like to see on the order of at least 30 molecules to start building predictive models that'll give you a predicted pose and binding affinity. But for eSim, all you need to know is I've got an active molecule in my hands, and you can go then and perform some virtual screens, and actually do quite well in terms of enriching for actives in your return pool.
Thank you very much. The next question you have is, for cases where you have partial overlap, can you use multiple ligands to patch together an overlap?
That's a great question, and it's something that's going to be discussed at some length in the multiple ligand alignment paper that we're going to be working on. It turns out in the sets of alignments that we generated for the benchmark, we explicitly wanted to look at that very question. So we made sure that for each N molecule alignment that came from crystallography, that the criterion for including that as a tractable alignment was not that all pairs of molecules overlap with each other, but that the union has good sort of union-wise overlap. So the answer is yes, that does work. It doesn't work all the time, and it's the most challenging, theoretically tractable case that we get. But yeah, you do see the ability to patch together over a larger binding site, partially overlapping ligands. I think there's a little bit of luck involved there. There's certainly some variability in performance, but long answer, but sort of yes.
Great. Well, sticking on the topic of multiple ligand alignments, there's a question that says, is there an optimal number of ligands for the multiple ligand alignment, some targets can have upwards of 200 ligands with known structures?
So what we typically do for large numbers of ligands is do it in stages, where we recommend to users to choose between 3 and 6 ligands that are reasonably active that have relatively different scaffolds. So try to find some diverse actives, generate a core alignment using the multiple ligand alignment approach in eSim3D, and then use that as the target then to align the remaining molecules that you have. Just as I showed in the benzodiazepine example, we used 4 molecules there to generate a core binding hypothesis, and then in just a few seconds we're able to align the remaining 150 or so to the core alignment. In the Kwanzaa approach, all of that is completely automated, and actually we're thinking about bringing that technology over into the eSim3D module as well, so that if you have a very large number of molecules that where you want to see a mutual ligand alignment, it'll automatically select a few from which to build a core hypothesis and then construct a full clique of all, say, 200, 300 molecules for you. It's already implemented. It's merely a matter of sort of copying the command line stuff over from one module to another. But it's not even hard to do right now in the existing E3D implementation.
Fantastic. Here's a provocative question. What would you classify as difficult cases for eSim?
Okay, so I think all methods have a slightly harder time when you've got molecules of very different sizes. So when you look at, for example, the carbonic anhydrase case, where you had a teeny tiny active ligand that you're using as the query, that is a particularly odd case because the warhead there in that case is extremely weird. And so we ended up doing very well in terms of the performance of the virtual screen, even though all of the other active ligands were quite a bit larger. In the general case, though, where you have ligands of very different size, that can be a little bit challenging. The eSim method is actually asymmetrical. You're asking a question about how well the subject ligand can mimic the parts of the target ligand that it can make itself look like. Vice versa is a different question. So because it's asymmetric in that sense, it works reasonably well, but that's still hard. The very worst case, though, again, which we consider technically intractable, is if you have 2 ligands of similar size, but which actually, when they're bound, only overlap a teeny tiny bit, say 10% or 20%, you're never going to get that right without other information. So that sort of drives the hardest case, where you get partial overlap between ligands that you just sort of can't know about until you see the answer.
Fantastic. And on a similar vein of challenging cases, how does your method do with covalent compounds?
We have done exactly 0 experimentation with covalent compounds. The way we would suggest people do that, though, typically when you've got some covalent inhibitors, you have a particular chemical functionality that allows the sort of covalent attachment to the protein, typically protein target in question. What you can do with eSim is provide a substructure-based positional constraint, so that that covalent thing will always end up in the same place. So then you can run eSim in a mode where it's constrained as tightly as you'd like or as loosely as you'd like to enforce that particular geometric constraint. So then the rest of the molecule that is not part of the covalent interaction will be aligned according to the optimal sort of eSim calculation, but the piece that's doing the chemistry with your biological target will be aligned very tightly. So we haven't done that. We haven't focused actually over the last 30 years on covalent targets very much, covalent ligands, but you can do it.
Thanks a lot. Well, the questions are still coming thick and fast, and we'll get through as many as we can in the remaining time. So here's a great one. Do you need to predict pKa of ionizable groups?
The ForceGen and FGen3D methods have a set of heuristics to protonate things at physiological pH. These are pretty good at picking out basic amines, carboxylates. There's a chelation flag you can set to deprotonate common things like hydroxamic acids and other known chelation moieties. I wouldn't say that the heuristics work very well when you get, say, a complicated heterocycle where substituents on the heterocycle might influence the pKa of a particular, nitrogen, for example. So if you look at methotrexate analogs or trimethylprim analogs where you see substitutions on the ring that are highly electronegative or the other way around, that affects the pKa of a nitrogen that is often protonated, which interacts with a carboxylate on DHFR. We don't get into that level of hair. I will point out, though, that StarDrop within the Optibrium set of modules has a pKa prediction module. Maybe Matt can talk about that for a second.
Yeah, absolutely. So many of you will know that within the ADME-QSAR module of StarDrop, we have our pKa model that uses semi-empirical quantum mechanical approaches to estimate the pKa of compounds, and very nicely handles sort of multi-protic compounds as well. We published that in Journal of Chemical Information and Modeling not very long ago, and if you'd like to know more, we've also got a dedicated webinar to that on our community website. So you're absolutely right. You could use that to look at the protonation states of more complex scenarios and ensure that you had the most appropriate protonation state for the alignment calculation. Definitely.
Yeah. So if you've decided that the pKa's and you have protonation state that you like, you can specify that to the sort of pipeline, either by being explicit about formal charges in the SMILES string themselves, or in an SDF file where the formal charge flags are set correctly. And then the eSim3D module can be told not to then heuristically reinterpret protonation, but just to use what you say. So you have the ability to deal with that, but inside the E3D module itself, protonation is fairly heuristic.
Fantastic. Here's a very specific question about SAR. In particular, the estrogen receptor example. You mentioned that one ligand sort of flips relative to the other in the binding site. Is there some directional hydrogen bonding, or what do you think is driving the difference?
It's a good question. I don't know how familiar all of you are with sort of ancient QSAR work, but way back in the late 80s, Dick Kramer published an example of testosterone binding globulin and a bunch of mostly naturally occurring steroids. And what you see is really interesting. So if you go from testosterone down to TBG, and then you shift over to estradiol, you actually see a left-right, top-to-bottom flip of the steroid backbone, completely counterintuitive. And it turns out that the hydrogen bonding, you can actually accomplish the hydrogen bonding using either orientation, the conventional scaffold-based one or the one that was proven out through crystallography. In the case of the estrogen receptor here, I think you can get both alignments to behave. And Matt, if you could let me share back my screen here, I can show this.
Yeah, absolutely. I've just handed control back to you.
Perfect. So I've got that slide up. Can you see that now?
Yep.
Okay. Yeah. So in this case, you can actually see that these ligands are actually functionalized with hydroxyls on both sides. It does turn out, though, that the counterintuitive flip is able to make a slightly better overlap in terms of the hydrophobic shape of the molecules, while still being able to present the two hydrogen bond acceptors that are critical in the right place. So it's surprising. What ends up happening with eSim is it predicts both orientations. I actually think that it predicts the correct one at a slightly higher score. But I think taking credit for that is probably wrong because the scores are fairly close. Taking credit for getting both alignments in the top 20 predicted, I think that's fair in this case, because these things really are quite sort of symmetric.
Wonderful. Gosh, they're still coming in. So is the molecular overlap reported out with the alignment results?
Well, that's a good question. That's not something that we've actually done. Hopefully somebody who has a pen handy will write that down, because it's actually a really nice idea. It's easy to calculate from the final poses, and showing quantitative molecular volumetric overlap would be kind of cute, I think, as one of the additional statistics that people could use. One thing that's sort of related to is that if you have an, say, extremely large 2 million, 200 million molecule database that you've run ForceGen on, so you have a preset SFDB, you can use overall volumetric ratio between your subject and your target molecule and your subject molecules to make things go much faster. So if you're looking for things that are very similar in size to your query molecule, you can set a volumetric range of 0.8 to 1.2, and that way you'll skip through a very large number of molecules that have a very different overall volume, and only focus on those that have a very similar volume. You can also bias the search in eSim to avoid volumetric extrusions where you'll stick a substituent out. It turns out that doesn't typically improve virtual screening performance, but it often makes people happier when they look at alignments to see that sort of the hands and legs of the subject molecule stayed inside the bus, so to speak. We're working more on trying to figure out when it makes sense and when it doesn't make sense to impose things like volumetric constraints and so forth, and doing virtual screens and also post prediction.
Yeah, that makes sense. So I realize we've just reached the top of the hour, and we're still got a fantastic audience. So thanks to everyone for sticking with us. I'll just go with one more question, and we'll obviously follow up with all of the others or any others that there are by email as soon as we can. But there's just one technical question here, which I thought was really interesting, which is, do you have any data on scaling with machines greater than 8 cores?
Yeah, so when we get up to 36 or 72 or larger numbers of cores, it doesn't scale up quite linearly because you still have to pay the input-output cost. You can control the number of molecules that get buffered in from an SFDB. So if you have a 2 million molecule SFDB, you can buffer in 10, 20,000 molecules at a time with a volumetric constraint and keep all of your cores pretty busy. So it's not quite linear with the number of cores, but it scales very well. And so you can use many core machines quite effectively. It's all implemented using OpenMP.
Fantastic. Well, thanks for addressing so many questions, AJ. That was absolutely fantastic. I just wanted to wrap up by just a reminder that we have another webinar hot on the heels on the 2nd of June. And this time it's actually a webinar being run by the Pistoia Alliance, and we were delighted to be invited along with Fabio Broccatelli from Genentech to talk about the kinase profiling project we worked on them with using the Alchemite deep learning imputation method. So we hope you'll be able to join us for that webinar. The link is here on the screen, but equally you can find that on our website and in our regular posts. And that just leaves me to say a very big thank you to AJ for a great presentation and really fantastic and fascinating results. Thank you all for joining us today. It's been great to see you in the audience. Just a reminder that the recording of this webinar will be available on our online community very soon now. And so if you'd like to refresh your memory or share this with any of your friends and colleagues, please feel free to send them a link. We will drop an email to you with details of where you can find that. And if you do have any questions, want to learn more about the eSim3D method, the upcoming new module within StarDrop or StarDrop itself, please do contact us at info@optibrium.com, and we'll be happy to answer your questions. So thank you all once again, and we look forward to seeing you again soon. Bye-bye.
Bye, man.